Speaker is Dr. K. Sreekumaran Nair. He's professor of medicine. He's a Dale Murdoch named professor of nutrition in the Mayo Clinic in Rochester. He's a University of Kerala graduate from 1973 and has traveled all over the world from AIMS to Auckland University to Charing Cross. His main research, and is known all over the world, is focused on aging, which deals with energy metabolism and the importance of altered protein turnover in diabetes and aging. He's a Mayo Distinguished Investigator. I won't take any further time. I will have Dr. Nair present his talk here.
Thank you. Thank you very much. Can you hear me and can you see my slides?
Please share the slides, Sri.
Oh, I thought I shared it. Okay, let me try again. Okay. Can you see it?
Yes. It's coming. Yep.
Okay. How about now?
Yep.
Okay.
Make
well,
it full screen.
Again, it's a huge pleasure to give this talk, and I would have preferred to give this talk facing all of you. This is a new culture. We all have to really get used to this. Well, I have a long-standing interest in aging. That's because I always consider my specialty in diabetes. Diabetes is an accelerated aging process. There's a lot of common between aging and type 2 diabetes. Here you can see that diabetes and pre-diabetes progressively increase with aging. And many of our research show that this is partly related to changes occurring in the skeletal muscle and also mitochondria. Well, there is a common pathologies, both the diabetes and aging affect. Aging affects, finally, the aging effect is mostly on these three post-mitotic tissues: brain, heart, and skeletal muscle. They are both post-mitotic tissue, which means they are produced, cells are produced once they don't replicate much, very low ability to replicate, which means they accumulate a lot of damage. And that is true with aging. And also these tissues are the one mainly affected by diabetes also. Of course, diabetes, there are a lot of macro and microvascular diseases. And aging, there's a decline in many hormones occurs. There's a lot of exciting data about senescent cells, especially the fat cells, which can secrete a lot of toxins, which can have major impact on all these tissues. And of course, sarcopenia and frailty. So aging is a modifying factor that accelerates many pathologies, causing decline in the quality of life and early mortality. So aging is unstoppable. We all know that. But the question is, can we slow down aging? That's what we are all interested. So we don't. The longest living human, as far as we know, is a woman in France who lived 122 years. So I don't think anybody in the foreseeable future can think about exceeding the life expectancy. And life expectancy, to some extent, is determined by the genes also. But our aim is to really have a what we call a healthy lifespan. And that's the idea of slowing down the aging process. Many years ago, we wanted to find out whether can we slow down aging or reverse some of the aging process by hormone replacement. You all know the story about estrogen replacement in women. And then of course, we were involved in some of the growth hormone replacement. Growth hormone can probably increase lean tissue in the body. But if you look at the performance of the muscle, for example, growth hormone has no beneficial effect. And give it sufficient amount of growth hormone to have effect on, functional effect, which can cause a lot of adverse effect. For example, carpal tunnel syndrome, many arthritis and joint problems, hypertrophy of the heart. So that's not an answer. So we wanted to see, there was so much literature showing that DHEA progressively decline with aging, and a lot of association has been shown between DHEA and all these three organs I was talking about. Then came some of the very interesting studies from France showing that replacing or giving DHA to rodents almost slow down their aging process, prevent them developing heart disease and also cancer and all. But one thing you have to remember is there's very low or not much DHA in these rodents. So it was basically supra-pharmacological DHA replacement. When we started this study, there's about $550 million industry online supplying DHA. In fact, when I went to one time give a talk in Karolinska Institute and also many other universities here, many distinguished people, older people who are taking DHA. That was the situation. So we want to see any science behind it and also testosterone replacement in men. So basically we replace the testosterone DHA in men and DHA in elderly women, and all of them, the DHA replacement brought to same level as the young people, and for 2 years. The result is absolutely no effect at all. Neither low dose testosterone or DHA has no effect. So that chapter is closed. And in fact, I mentioned about this industry of $500 million industry, which is now just over $5 million industry. Some days negative findings have some major impact. This is showing the MR scan of MR imaging of calf muscle. I had better image. I took it because this young woman and this older woman, both of them have almost same body mass index. What you can see is that here, this older woman, even though the circumference of the calf is almost the same, the muscle is substantially replaced by the fat mass. So there is a change in the composition. This is very important. Remember, BMI is often used as a measure of obesity and also its effect on insulin sensitivity. With aging, BMI probably has no value because you can see that body composition change, fat tissue substantially increase. Well, this is a study many years ago we did in Rochester, Mayo, and what you can see is we took people from every decades and measure their maximum aerobic capacity, VO2 max. Normally, if you look at the VO2 max, there is women have lower VO2 max than men. But if you normalize for the fat-free mass, there is no difference. Here, what I'm showing is when a fat-free mass normalized by the fat-free mass, still there's a progressive decline in the VO2 max occurred related to age. Same thing true with the muscle mass. Even after normalizing for the muscle mass in the leg, there is a progressive decline. So age has an effect on your VO2 max and muscle mass. The question is it reversible? One of the area I focus for many years is this is started with my early studies in type 1 diabetes. We found that insulin has a major role in regulating mitochondria. So mitochondria, as you know, is this organelle. This is the EM from a muscle biopsy we had done. This actually a bacteria got into the eukaryotic cell several millions of years ago, but nucleus took control of it. Make this bacteria to really do one of the most important role in physiology, which is really burning the fuel and converting the carbohydrate, fat, and protein into ATP. But in that process, even if fat is oxidized or beta oxidation occurs or carbohydrate is oxidized, if there is an inefficiency in the oxidative phosphorylation, it does not produce enough ATP. In fact, it can produce a lot of reactive oxidant species. If those reactive oxidant species, they are not buffered by antioxidant defense system, the result is the reactive oxidant species can damage protein, and the protein can accumulate. Aggregationist protein can occur in many organs, including the brain, contributing to degenerative brain disease and many other problems. It can be toxic to the tissue. So mitochondrial function is important. We have reported many years ago that if you look at the mitochondrial protein synthesis, there is a decline occurring young to middle age. And then onwards, not much happening, in fact. There is a decline in the mitochondria biogenesis occurs. I'm now showing the data of the transcript level and the translation and everything. And we also look at this oxidative enzymes, which are mitochondrial oxidative enzyme. Mainly citrate synthase is main involved in the citric acid cycle pathway, and cytochrome C oxidase in the electron chain transport needed for ATP production. Both of them, there is an age-related decline occur. Later on, we had the opportunity, later we reported this in again the PNAS, showing that mitochondrial DNA abundance decline. This young, middle age and old age, mitochondrial DNA abundance progressively decline. And this seems to be associated with an increase in the oxidative damage to the DNA, which suggests that in this oxidative damage, DNA is a measure of damage occurring due to reactive oxygen species. And there is a progressive decline in the maximum ATP production capacity. They are almost parallel. So your ability to produce ATP is reduced. ATP, as you know, is critically important for our cellular functions. That can contribute to age-related decline in many of the cellular functions. So we wanted to see whether this is really related to aging, and that causes insulin resistance, as I mentioned. But if you look at the young and old people, if you look at their VO2 max, you can see that young, lean, young, older people, both of them have decline on the VO2 max, whereas young, lean, and obese lean don't have any change in the VO2 max. So it doesn't seem to be a function of the insulin sensitivity. Here I am showing the ATP production, which is really, this is using many of the substrates. We can focus on one here. There is definitely an age-related decline of the ATP production occurring. And if you look at the insulin sensitivity by using the classic hyperinsulinic euglycemic clamp, that means rate of insulin infusion needed to maintain the glucose at the same level while the same amount of insulin is infused. Here you can see that in all this group, glucose is maintained. This is young lean, young obese. This elderly lean, elderly obese. You can see that obesity or accumulation of the fat reduces insulin sensitivity. It's not the aging which is causing it. Aging can reduce the ATP production, but aging doesn't reduce insulin sensitivity. So there is this dogma that when you get older, you become insulin resistant. That's not entirely true. If you don't accumulate fat, that doesn't happen. Moreover, we have looked at people who are regularly exercising for more than 4 years by biking long distance bikers. And so we brought some young trained people who are doing more than 4 years, old trained people, and young sedentary people who don't do that, old sedentary people. They are all healthy. What we are seeing here is the glucose infusion rate here, which means higher means you are more insulin sensitive. And these two groups are young trained and old trained. This young sedentary and old sedentary. What it tells you is that the insulin sensitivity is not related to whether you are young or old. It's all related to your exercise habit. Those who are sedentary, they develop insulin resistance. So there are two things we learned from that. Insulin resistance at the age can occur because you are accumulating fat. As you become older, you may lose your lean tissue, you maintain the same food habit, your metabolic rate go down, you accumulate energy as fat, which can contribute. Second thing is you become sedentary, and that can cause insulin resistance, increase the chance of becoming diabetic and other problems. So we want to look at really, that was a cross-sectional study. What happened if you, in fact, if you exercise people 3 different ways, by resistance exercise, aerobic exercise, and also by combined exercise program. And here you can see the glucose infusion rate. Here what we are showing is the change occurring. This is a 0 baseline change occurring. You can see that this is, we performed a high intensity interval training, which is a very effective aerobic training. This is a high intensity resistance training. This is a combined training, which is a sort of a less intensity than these two, but a combined training. You can see that in everybody, especially young people, all type of exercise program improve insulin sensitivity. Doesn't matter whether you do aerobic or resistant or combined training. Older people in the combined training did not do that well. There is evidence we have and others have shown that older people, the intensity of exercise has to be higher to reverse their insulin resistance as opposed to young people. So the next question we asked is, how about VO2 max? You can see that the VO2 max increase HIT, the young and old people. Young people have substantially more increase than older people. and resistance training did not do anything. Okay, and combined training increased. So you need to do aerobic exercise component. Resistance doesn't improve your VO2 max. I'll show you later. VO2 max is a good index of longevity, or rather, a poor VO2 max is a predictor of premature death. And when you look at the mitochondrial function, yes, aerobic exercise improved, resistance did not do that. If there is an aerobic component, to improve that. It shows that combined training can improve both the VO2 max, muscle strength, and also insulin sensitivity, provided you have sufficient intensity for doing it. Well, how about the muscle strength? High intensity interval training, which increased the lean tissue, you can see that it's a fat-free mass increase, but did not increase the muscle strength. Look at the muscle strength increase. Young and old people, substantial increase in muscle strength occur. And resistance exercise, they are very responsive to resistance exercise. And combined training also improve the muscle strength. And fat-free mass increase whenever there is a resistance training occurring. So that way, the conclusion from this study is that aerobic exercise improved insulin sensitivity and mitochondrial function and VO2 max, but resistance exercise also improve insulin sensitivity. But unlike other exercise, aerobic exercise improve the muscle strength and muscle fat-free mass also. So you need to do both. Well, we are trying to understand what is causing the insulin sensitivity. We know that based on our studies and also studies done by others, aerobic training, the Bruce Spiegelman has discovered this PGS1 alpha. We have collaborated with him to show that the aerobic increase PGS1 alpha 1 on isoform, resistant increase another isoform called PGS1 alpha 4. One is more of a oxidative phenotype that causes mitochondrial biogenesis. That decline in mitochondria occurred with aging, sedentary older people can be almost completely reversed by aerobic exercise program, especially high intensity. And oxidative metabolism increase. The result is that mitochondrial efficiency improves, less reactive oxygen species occurred, the oxidative damage reduces. And also reactive oxygen species also contribute to insulin resistance. And of course, your ability to really metabolize glucose and fatty acids substantially increase. Resistance, this PGS1 alpha 4, we have a paper in Cell with Bruce Spiegelman and all showing that PPR beta, combined with PPR beta, increase muscle hypertrophy. The question we are addressing now is currently one of the NIH, newly NIH funded grant. We are looking whether PGS1 alpha 4 through PPR beta can also increase the glycolysis. You know, the aerobic training, you can go through oxidative metabolism, get enough ATP to do that. The problem with the resistance training is you need immediately large amount of ATP, and glycolysis can produce it. That's our hypothesis, and we are investigating that. Well, this is, I just want to show that, many of you are familiar with this MET, metabolic equivalent of training, which is mostly VO2 max. So this study published in New England Journal of Medicine some time ago shows that if you look at diabetes, hypertension, smoking, obesity, high cholesterol, in all these cases, the relative risk of death is higher in people who have low MET or low VO2 max, and lowest in people who have the higher VO2 max. And this is now shown in many studies. Probably the best predictor of mortality is your fitness. In fact, the prospective study done by NIH in Baltimore showed that the one clear predictor of mortality is really activity. When you slow down, you can predict you are dying. It is also clear from studies done in C. elegans, mouse, monkeys, everywhere. When they slow down, you can almost casually predict they are going to die. So the important is keep acting. Well, another thing is that the new guideline of by American Academy of Neurology is that the exercise for memory and thinking, improve memory in people with a mild cognitive impairment clearly has been shown. In fact, there is no drug we have discovered so far which can really prevent the decline of brain functions in older people and also people with the diabetes. I must say that this is probably showing that diabetes increase with age, Alzheimer's disease has increased with age. If you are a type 2 diabetic, you have 2 to 4 times higher chance of developing Alzheimer's disease than others. So the combination of aging significantly increase your chance of developing a dementia. This is showing that you can really look at the brain and also look at the glucose uptake in the brain. And you can see the normal brain, minimum cognitive deficiency, Alzheimer's disease, a progressive decline occurring. So brain metabolism is declining with age. And one of the things personally thinking was that many years ago, I had done in collaboration with Professor John Warren in Karolinska Institute. John catheterized the internal jugular vein and also artery, and we really found out the exchange of various substrate across the brain. And we found that insulin increase in the lower has no effect on glucose uptake in the brain. Glucose, if you lower, still brain takes the same amount of glucose. That really we thought that the brain doesn't need insulin for the glucose metabolism. But this is a map really drawn by Ron Kahn's group in Harvard, Joslin Clinic. Ron and his colleague have shown that insulin receptors are there in the brain, certain areas highly rich, and also IRS1, which is one of the insulin receptor substrates, both are important for insulin action. But they are in certain areas of the brain, mainly cortex, hippocampus, hypothalamus, not much in the midbrain or various other areas or the posterior part of the brain. So they are in certain part of the brain. Interesting thing is that if you look at where the Alzheimer's develop, these are all the areas where the insulin receptors are rich. And if you really put that together with the fact that Alzheimer's is significantly higher, dementia is significantly higher in type 2 diabetes, there's insulin resistant. It is also higher in type 1 diabetes patient. So we have done a study as initial human study where we put these people through a high intensity interval training program.
Dr. Nair, you have 4 minutes.
Okay. I think I started late. Okay. Okay. The left parietal, left temporal, right cardiac, all these regions of the brain are rich in insulin receptors and also affected by Alzheimer's. 3 months doing a PET scanning, you can see that the glucose uptake increase in all these areas. This is not Alzheimer's people. People who are young and old, who are healthy, showing that exercise can increase glucose uptake in the brain, that means brain metabolism. And this is, I'm showing that when you give intranasal insulin, this is a study in mouse we have done, we recently published. You can see the stage 3 respiration increase when you give intranasal insulin, ATP production increase, and mitochondrial DNA copy number in the brain increase. These are all areas, hippocampus and areas where memory is involved, and PGC1 alpha increase, all of them showing insulin can increase mitochondrial biogenesis in the brain. We put this mouse on a high-fat diet, which really you can see that the AKT phosphorylation and many other markers of insulin signaling, they all declined, or rather they don't move up with insulin. And when you really put them through sedentary mouse and put through a, sedentary mouse, you can see the insulin signaling go away, but high-fat diet reduces that. If you exercise them, that return to normal. And also, I just want to move from there because the time ATP production decline in the high-fat diet induced mice, and this increases on exercise. And also showing that the reactive oxygen species production increases, and you can reduce by exercise. And here I want to show the EM scan of the mitochondria. This is a sedentary mouse. This is high-fat induced mouse. Mitochondria is completely disrupted. The exercise makes them normal. So again, if you look at the brain protein, you can see that this is showing that all the red ones showing the mitochondrial protein, they are showing that they are all, this is showing more oxidation occurring. And they are all increasingly oxidized when you give high-fat diet. This is showing that that's significantly reduced by exercise program. This accumulation of this oxidative damaged protein accumulate in degenerative brain disease. Again, showing that it's not only muscle, but also in brain, exercise will benefit. So aerobic exercise training increases brain volume, which has been shown by others, and microcirculation of aging human, especially microcirculation in the hippocampus and areas involved in the memory. Blood factors transfer, this is a recent study came in Science. If you exercise and mouse and transfer their plasma into this sedentary mouse, that seems to increase the neurogenesis and cognition in aging brain of the mouse. And again, recently shown that neural adaptation to strength training, which I can briefly say that if you really resistance exercise increase the connectivity between different parts of the brain, and in fact enhances the areas of the brain involved in the alertness. So I'm going to show this slide at the end. There is no substitute to exercise. The exercise can do a lot of things. We are all trying to find out the pathways it's doing it, with a hope that we can target those pathways, really develop some molecules and drugs which can be used for people who cannot exercise. But as it is, there is nothing. Your voluntary exercise is cognitively controlled. You had to do it. So with aging, a lot of people can become slower. If you voluntarily exercise, that improves the mitochondrial biogenesis and ATP production. And there is evidence that can increase the hypothalamic centers in the areas of brain, which increases spontaneous activity. When your spontaneous activity decline, slow down, you are going to the common destiny, you all have death. So that way, there is a strong case for, it's never too late. Even though aging process starts from the age of 30 years, really any age you can reverse some of the changes occurring. Many studies we have shown demonstrated that. I'll stop it there, and I want to thank many people who have done this study. This lot of people in the lab who has done it, and I'm not going to go through that. And we could not have done with the support of the Mayo Clinic, and also a couple of NIH Institute, NIDDK, National Institute of Diabetes and Kidney Disease, and National Institute of Aging. Okay, I'll be happy if there's any time to answer any questions.